Monitoring telomere length at telomere-by-telomere resolution, the method and clinically oriented applications, by Kathleen Collins, PhD. Dr. Kathleen Collins is a professor of molecular and cell biology at the University of California, Berkeley. Her group has led the way in reconstituting recombinant minimal telomerase complexes for studies of enzyme structure and mechanism, and in defining the composition, assembly, and regulation of the larger biologically active telomerase holoenzymes. Her group made the first connection between human genetic variation in telomerase, subunit genes, premature telomere shortening, and inherent syndromes of bone marrow failure. This continued research demonstrates the significance of individual differences in telomere length on the human health span. It's my pleasure to introduce Dr. Kathleen Collins.
Let's find the right one. Red. I guess we're going with red. Well, I want to thank you all for saying to listen to something a little different, because I'm going to tell you about new ideas for what I think is a greatly useful future diagnostic test. So I'm going to tell you about telomere length. And before I begin, I'm going to give you a very brief introduction to the telomeres themselves. This work that I'll tell you about has been done by a large field of scholars, including my group at the University of California at Berkeley for the last 20 years. You know that we have 46 chromosomes that contain all of our genetic code, the DNA in 46 units. And this slide shows you that DNA stained in light blue. I'm going to use a pointer on this screen, sorry. There's only a red one. Here, actually it's in a cell that is, thank you, much better, that is just about to divide. So there are 46 of these chromosomes. There's 2 copies of each one. And for many years, including my own undergraduate, we studied a lot of the light blue stuff. It was completely unclear what was present at the very ends of each of these linear chromosomes. The information to find that out was missing. But in fact, in the late 80s, there was a breakthrough. And now in a textbook picture, you'll see a structure like this, in dark blue, reveals a hybridization of a simple sequence T2AG3 running 5' to 3' towards the end. The ends of each chromosome are the same. They're an array of these simple sequence repeats, not a protein coding gene, an entirely different kind of gene DNA segment. These telomeric repeats are critical for the stability of the chromosome ends. If something like irradiation breaks off this end, the lonely end will go hunting among the other 45 chromosomes trying to steal. So, in fact, the loss of these ends is hugely deleterious for genome stability. So it was a shock when people went to measure the length of this buffer zone, the length of the protective telomeric repeats. So here, short to long, in cells grown in culture. And in fact, if you take cells from a very young person, say a 20-year-old, and you follow the number here of telomere length chromosomes, number of chromosomes with a certain telomere length, you'll see that all these telomeres are very long. But if you take the same cells from the skin of me, you'll notice that the telomeres are shorter. And in fact, if you wait 10 more years and you do the same thing, they're going to be even shorter. Now, this was a shock, because these ends are very critical. And yet it's clear that with age, in humans like us, our somatic tissues are not maintaining them. Now, we do have an enzyme that adds back these missing repeats, but it's only present in a small fraction of human cell types. I'm very briefly going to tell you what that enzyme is. It's called Telomere Ace, because I'll use that enzyme name, telomere synthetase, telomerase. And what's fascinating about telomerase, this is supposed to be here, is that it's composed of proteins and RNA. So this is ribonucleic protein, or RNP enzyme. So its rules are very different from the normal metabolic enzymes we think about, which is why we've been studying it. And what it does is break the central dogma. It comes to DNA, and it adds more DNA by copying an RNA template within it. Central dogma says DNA copies to DNA, DNA gives RNA, gives protein. This enzyme breaks that dogma, and it does so in order to add back these simple sequence repeats that are lost, necessarily lost with each round of cell replication, due just to the inherent requirements of DNA synthesis. So if you want to read more about telomerase, you can. We're discovering that it's an incredibly complicated enzyme with many subunits and a lot of regulation. But the best place, in fact, to read about telomerase and telomeres and their discovery are the Nobel Prize speeches, in fact, of the 2009 winners in physiology and medicine, Liz Blackburn, Carol Greiter, and Jack Szostak, who showed the presence of these simple sequence repeats and the enzyme telomerase and the critical nature of both of these for human cell maintenance. And these Nobel Prize speeches are generally good for historical context. I highly recommend them. But what I'm going to tell you about today for the rest of my talk are 3 points in decreasing concreteness of evidence. I'm going to tell you why we should care about monitoring and eventually manipulating human telomere length. This is very clear. We should care. I'm going to tell you about methods for monitoring telomere length. We have many good methods, and they're actually getting very much better. I think very shortly there'll be concrete clarity in how to do this. And then I'm going to tell you at the very end about applications of measuring telomere length. And this is where you all come in. Because academics like myself, we can speculate. But people who are providing the primary care are the ones that are going to have to think of all the different ways in which this will be useful for their patients. And I'll give you a few examples that my research has helped to understand. So why we should care about monitoring telomere lengths. In us, it's fascinating to think that mutation is constant. And the way that evolution has made sure that each generation of us gets the fewest mutations, gets the least damage, is by setting aside very early on in development from the fertilized egg, setting aside some cells, we'll call them germline cells, egg and the sperm, that divide very little. So that when a mother goes to give her germline cells to her daughter, in fact, there's been very little cell replication, very little loss of telomere. But the cells that determine our phenotype, that give rise to our health, are actually separate. We call them somatic cells. They branch off and they divide a lot. And it's these cells that maintain our tissues. So you can see that something like telomeres are really critical to maintain across the germline. But in fact, in our body, since our cells are not going to the next generation, they're not so critical. So this is the disposable soma theory of aging. It's very clear that the maintenance of somatic cells is different than germline cells. And telomerase is very active in maintaining telomere length in germline cells, and not so much so in somatic cells. So these cells don't have to be immortal. They don't need their genome to be perfectly fixed. We study this kind of process both using single-celled organisms, where each daughter is a mother, and in these cases, as a function of increasing cell divisions, telomere length is maintained, and this Telomere Ace enzyme is active constitutively. But if you survey cells from multicellular eukaryotes, this is a picture of human fibroblasts, as a function of cell division here, the story is very different. Most somatic cells, if you sample them, of increasing culture time, number of doublings in a dish, or with chronological age in the population, they're shorter telomeres, to the point where telomeres become so short that in fact the cell's genetic code is not stable anymore. This instability is one progression for cancer cells. But I want to focus on normal somatic cells, here on healthy tissue, because there have been some examples where a somatic cell lineage, like a progenitor hematopoietic cell, can transiently turn on telomerase and gain a little telomere length. And this will offset the rate of shortening and give that cell lineage more replicative capacity. So short telomeres are very deleterious. This slide has a lot of detail on it. You can read about the details of it if you're interested in genetic stability. But the point here is that short telomeres in different cell types actually trigger different types of program. Many times they arrest the proliferation of the cell. The cells will kill themselves, often. They will die, and they can convert to cancer. So all of these responses to short telomeres are rather unfortunate. And if a cell has telomere shortening as a function of cell division and acquires short telomeres, and only then activates telomerase, a mechanism to maintain those telomeres, the cell that grows out of this has genome rearrangements, transformed phenotypes. It grows very well. It's a cancer. Now that it has telomerase, it maintains telomeres and can just keep growing. But this is not a fortunate way to stimulate cell growth, because the short telomeres have allowed the cell to change character. So the real trick will be to monitor telomere length. And before telomeres have shortened to the point where they're inducing damage, here to develop ways to activate telomerase. Because if you do this experimentally in the lab, the cells that grow out, now they can maintain their telomeres. They can grow forever, but they're still very normal in physiology. So the consequences of activating early and activating late are very different. So these consequences of short telomeres aren't limited to just cancer. Even if no cancer grows out of the short telomere cell, because the telomeres are short and the cell can't continue dividing, there's a loss of renewal capacity. There's a failure of stem cell niches. All of these have been documented. Metabolic dysfunction. And in fact, those cells with short telomeres don't just subtract themselves from the population. They actively secrete cytokines and other molecules that are pro-inflammatory and are just not good for the local cellular environment. So these short telomeres are a real toxin for us. Good news is that in mouse models, it's very clear that if you give an adult mouse, hopefully one day an adult human, a little boost of telomerase, here through a gene therapy approach, hopefully in us through a small molecule, that it can increase longevity, increase tissue function, and not promote cancer, because you've activated the telomerase and the telomere maintenance pathway early enough that the cell physiology is still normal. So this was a very brief introduction to why we care about monitoring telomere length and why we should care, potentially, about manipulating it clinically. But in order to do that, there of course need to be methods for monitoring telomere length in humans instead of mice. And so I'm going to spend some time telling you about different methods for monitoring telomere length and where we have to go with these. So telomeres, of course, are DNA repeats. So there are ways in which we can monitor them based on their sequence, T2AG3. And you don't have to pay attention to all these titles. There's a color code. In black are, here in the late 80s, are molecular biology methods that we first used to measure telomere length that are rather time intensive and require a bit of twist of the wrist specialized knowledge. But there are also methods to monitor telomeres that are cytological, that are by fluorescence. You can hybridize a fluorescent probe to a whole field of cells and monitor telomeres in that manner. There also have been, more recently, some methods for monitoring telomeres that are using PCR, where you extract DNA from a bunch of cells, and then you try to amplify by PCR telomeric repeats. So these are three main strategies, and I'm going to briefly show you them so that you can understand why it's really critical to pick a certain type of telomere measurement when we're considering evaluating telomeres as a diagnostic marker. So the first assay, telomere restriction fragment, or TRF, this is what I was saying was our molecular biology method. What you're looking at here is a gel where small DNA fragments are at the bottom and large DNA fragments are at the top. And what we've done here, we study in my lab, we take cells and we let them divide, and at different numbers of cell divisions here, we harvest the DNA and we probe for the presence of the telomeres so we can see how long they are. And what you can see here is that if we take telomerase and by gene therapy we put it back in these cells and we measure telomere length, it's going up. Even as these cells divide, they're gaining telomere. But if we don't do that, here, you probably can't even see, but these telomeres are shortening. And these cells stopped growing right here. So without telomerase, these cells grow and they all stop right here. Telomeres are too short. But if you add telomerase back in here, they will just keep growing and keep growing and keep growing and maintain their telomeres. So this is an early kind of test we used for cells in the lab to ask how long or short the telomeres were. And this was very useful in our early studies of the linkage of short telomeres to disease, because we were studying a disease, a bone marrow failure, dyskeratosis congenita. And one of the subunits of the telomerase enzyme that we had been studying turned out to be mutant in this disease. So we could ask, well, if we take carriers of the disease, for example, the mother here, and we look at her telomeres by this TRF assay, and we look at her son's telomeres, who's much younger but is affected with the bone marrow failure, you can see that his telomeres are much shorter in blood cells. And this is not normal, because if you're younger, your telomeres should be longer. You should lose telomere length as you age. But here, this person has clearly suffered premature telomere loss. And these telomeres are, in fact, at the length where they prevent more cell division, which is why this patient has bone marrow failure. They have just run out of proliferative capacity. They have run out of telomere reserve. And they've run out early because they inherited a gene that turned down telomerase. So they inherited very premature bone marrow failure, aplastic anemia, other individuals that inherit telomerase deficiency, pulmonary fibrosis, liver disease, many other phenotypes of limited cellular renewal. So this method was fine for establishing that short telomeres were useful to examine. But in fact, it's very labor intensive. And so there have been subsequent methods that are intended to be more general and to potentially use in a clinic. So this TRF assay is limited. The first type of assay that was developed was a flow cytometry coupled to fluorescence in situ hybridization, in other words, a fluorescence probe against the telomere. And in this flow fish method, the cells are disaggregated, hybridized with the probe, and then on a per cell basis, the amount of fluorescence is counted in that cell. So if the cell has long telomeres, there'll be more fluorescence. Less telomere, less fluorescence. So this data is complicated, and all you should look at is the red dots. I'm going to explain to you what's on this graph. If you survey with increasing age here, the telomere length in different subtypes of cells in your blood, for example, let's do lymphocytes. Within these black curves, all these open circles, those are normal, quote-unquote, individuals. And their telomere lengths decrease with age in these blood cells. You can see there's a huge population variation, and there is really. We inherit, and the environment forces us into a huge population variation in telomere length. But what you can see are the red symbols here. They're all off the bottom of the curve. So very young, in teens even, patients with dyskeratosis congenita, this bone marrow failure, they wind up with such short telomeres that there isn't any more replicative capacity left. So studies like this, using different cell types of the blood, have shown that every dyskeratosis congenita patient has extremely and prematurely short telomeres. And now this is diagnostic for this disease. Different kinds of bone marrow failure, only some of them are caused by this telomerase deficiency. How you treat this disease when there's telomerase deficiency should be very different. You can't just give this person a stimulant of hematopoietic stem cell division. They don't have any stem cells left. Okay? So bone marrow transplant would be the option. So this is an example of flowfish. But in fact, there's a limitation of flowfish too. Because remember, every cell has many, many chromosomes and many telomeres. And studies suggest that it's not the average length of telomeres that will determine when a cell stops dividing. In fact, it's only the shortest telomere. So in a cell, if a few telomeres are very short, the cell has to stop growing. So we need to know not the average telomere length in a cell. We need to know, in fact, whether the cells are coming close to having any short telomeres. So there are additional methods for that now use not whole cells, but what you're looking at here again is what I started with, a spread of condensed chromosomes from a human cell. And you can see perhaps at the end of the blue, which is the DNA, these red dots. And you can see that if you do this fluorescence in situ hybridization, this fish, on a chromosome spread, you can see each telomere. And you can quantify this red signal at the end of each, and you can know whether there are any short telomeres. But this also is very time intensive, you can imagine, and it only works for cells that are dividing, because you can only have these condensed chromosomes during the process of cell division. It's a metaphase spread. So there have been some translations of this method to tissue sections here. You can see the DNA marking each different cell in blue, and there are some red signals in here that are the monitor for telomere length. So there's some success, in fact, in using a tissue section to look at telomere length. This is particularly useful, for example, if you're looking for the presence of cancer, because the cancer cells have very short telomeres. And if a few of them are mixed in with the normal tissue, you might pick up one cell that has very short telomeres in a field of cells that have normal telomeres. But in fact, this is also rather technically laborious. So we're in the field trying very hard to move to reliable, high throughput methods for measuring telomere length that can be clinically useful. And some of these methods are summarized. They'll send you to the literature. But I'm going to point out on this slide that in addition to the low throughput assays, like that telomeric restriction fragment plot I started with, there are also some high throughput assays already. And in addition to assays that measure average telomere length, like that flowfish that I told you about, average telomere length per cell, or in a whole population of cells getting an average by PCR, these are less informative than methods that will tell you the percentage of short telomeres in a population of cells or even in a single cell. Because single cell by single cell, if we know profiles of telomere length, we'll have the complete information about what different types of cell populations are present, as well as the proliferative capacity in each of those types of cell population. If you want to talk more about this, I encourage you to find me in the exhibitor booth later this afternoon. So there are methods that use single cells to do fluorescence in situ hybridization, some of which are high throughput. So I'm just going to show you one example of that, where we are now, which is to be able to take cells. Right now, they have to be cells that are separated from each other and spread into the bottom of a microtiter plate, where each cell being separated can then be hybridized with a probe to reveal the amount of telomere per cell and even to see different telomeres. Of course, this has to be read computationally instead of manually, and the data processing will then give a graph for telomere length distribution here, number of cells at each telomere length, that can be used to classify different populations of cells, for example in the blood, or different telomere lengths within a single cell. And we're just at the point now where this methodology is becoming reasonable to use on a much greater scale. So I've promised you that we're getting there with the methods for monitoring telomere length. There are assays for telomere length. You can have your patients sample. And in the next year or two, these will become very concrete. So I hope I've convinced you we should monitor telomere length. I hope that I convince you that we will be able to monitor telomere length. So now I want to end by talking about some specific examples that I can come up with for what uses, what specific applications of telomere length monitoring you would want to consider. And here's where I encourage you to think of your own. So the first, I think, real application are genetically linked diseases where you know, for example, that your patient might be at risk of short telomeres. So I'm going to explain this. Telomere length decreases as a function of age in all of us. And with a diversity of rates in the normal human population, this line here is drawn as a line. But in fact, this is a huge cone. Because within the normal human population, there's a great range of telomere lengths, beginning, and in fact, a range of loss rates. So this line here hits the disease risk purple section at 70 to 80. That's just an estimate. That's my speculation. This range is actually much greater. So superimposed on the normal range are genetic predispositions to premature telomere shortening that we can call telomeropathies, or telomeropathies. And in fact, in this population, telomeres may short telomeres may be present from birth, in fact, but also the rate of telomere shortening can be enhanced relative to the normal population, and therefore disease risk develops much earlier. So this is clear because we have shown that in these bone marrow failure patients with dyskeratosis congenita, they inherit low telomerase activation. They start life with short telomeres and they lose them faster. So there are clearly patients that we've characterized who are suffering, who have hit the disease risk because of their short telomeres. There are many other cases of this, but in fact, even if the primary genetic defect isn't telomerase, there may be other diseases that come with a risk of higher cell turnover or higher telomere loss due to DNA damaging agents. And so I think there's a lot to learn about that category. And what I'll show you here is a simple way that one can test for that, a diagnostic. This is, again, the lymphocytes with age versus telomere length for the normal population in open squares, and for patients in red with dyskeratosis congenita. And you can see that telomere length of this size when you're below 20 is pretty diagnostic for this disease, distinct from the normal population. So there are clear diagnostic cases already, but people have begun now. This is a paper just from this year in the high-profile journal Cell, where certain people, for example, Michael Snyder at nearby Stanford, decided to omics himself as a preventative medicine. He sequenced his own genome, and in doing that, he looked for disease genes. And he is a carrier for a mutant TERT, which is one of the subunits of telomerase. So he was worried, and he should have been worried. He had one good copy, one bad copy. He was heterozygous. So in fact, his telomeres were okay. He had one of these telomere tests from a commercial company, and so he knows not to fear too much, but it would be useful to know if your genetic background puts you at risk for having short telomeres, in the same way that he knew to ask whether he was at risk for having short telomeres because he had done genome profiling. So in addition to the whole genome prediction profiling, there are other cases where you would want to know, I think, whether your patient had short telomeres. So for example, this is just a distribution from a publication of the association of very short telomeres with breast cancer patients. So what you'll notice is that these BRCA1 and BRCA2, in fact, all the BRCA disease carriers, who have breast cancer due to this genetic inheritance, they happen also to have very short telomeres. So among these patients, more of them have very short telomeres. What this means is that the way in which this subset of breast cancer patients have gotten the cancer is different. We all know this, right? Not every breast cancer is the same. But these genes, these BRCA genes, are important for DNA repair. So without this particular set of genes, each damage to the cell results in more telomere shortening. So these patients, in fact, have shorter telomeres. And this, again, is something you should be aware of when you go to think about therapy. Because these cancer cells will have short telomeres, but the surrounding tissue will also probably have short telomeres. And it doesn't have a whole lot of proliferative potential left. So if you want to ablate a large cell population, you may not expect it to grow back. In addition to these genetic factors, there are a whole lot of environmental factors that relate to telomere shortening. And this is where we're just beginning to appreciate the complexity. Because the number of times you have to divide a blood cell may not change the genotype. The gene deficiency is still there or not. But the telomere array is shorter every time the cell divides. So if there's damage to a cell tissue, and the tissue renews, and the tax on the renewal is higher in your patient than in someone else, in fact, there's an increased rate of telomere shortening with age. And this increased rate of shortening here has been associated to stress, poor diet, inflammation, viral infection is enormous, like this, because the virus is killing a lot of cells, and the turnover rate that's imposed by that cell death and renewal is enormous, such that the age at which disease risk begins is much lower than the normal cell population, although you're normal. So I think that here there's a lot to understand about this, and again, I'll just point you to the literature. There are many reviews now of the correlation between diet, nutrition, and telomere length, and between lifestyle factors and telomere length. And there's big grids, and we're just now beginning to be able to predict what, in particular, which types of environmental differences have the most impact on telomere length. I think this is an up-and-coming topic, and I would encourage you, if you have any research interest in this, to get involved, because there's a lot of important things to learn. So I'm here on this slide. This is a summary for you of where I think the telomere science can contribute. We're really coming into an era, you know better than me, where we need personalized medicine, and we need preventive medicine. Because if you can treat a disease before it starts, the patient's health is greater and there are fewer complications. And this is particularly true in the case of telomeres, where the consequence of waiting for short telomeres is very different than treating pathology before the telomeres are too short. If you wait for short telomeres, you get cancer. If you don't, you won't. So there are inherited factors that lead into choosing a personalized preventive medicine. There are many genetic tests. I've had them done on myself. You can ask about risk factors for disease. But in fact, that's not sufficient information, I would argue. Because your genetic composition, the genes, the proteins that are synthesized, the enzymes that are there, are only part of your genetic makeup. There are also telomeres. These buffer zones on the end of the chromosomes, they're critical. They're not coding for proteins, but they're an element of the chromosomes that's essential for understanding cellular health. So understanding telomeres, there's both a component that's inherited. You can inherit a deficiency in maintaining those telomeres. But there's a huge, I think very huge, environmental component. We don't even know all of the exposures that lead to telomere shortening. The good news, though, is that the inherited component and the environment component, you don't need to measure them separately. They're both acting to give you a certain number of telomeric repeats, and the damage that's been done, the amount of proliferative capacity you have left, is a simple readout in the length of the telomere. It's like a genetic test. You're just asking about the DNA content of a person. But it's unlike a genetic test in that different cell types and different chromosomes in the same cell have different telomeres. Not every cell has the same gene, right? So it makes telomere testing a little different than a genetic test in that you have to take into account what types of cells you're looking at, the health of those cells, and the profiling of telomeres within those cells. But at the core, it's the same. It's a DNA test. And so it's doable. And the combination of these two are going to be particularly powerful. Because if you know where your telomere length stands, and you know from genetic factors whether you're at risk for losing telomere length faster, you can then have a more powerful prediction of disease risk and the time of onset. And I think that we can also use these predictions to develop new treatments, and in fact to monitor disease treatments to see what is stabilizing the population of cells through telomere length. So of course aging is the highest risk factor for all diseases, and telomere length is a hallmark of aging. So I have 4 minutes left, and I have 2 slides. On this slide, I'm just summarizing the current utilities of telomere length determination. One already in practice is to identify patients with inherited telomeropathies, told you about that, and to ask about healthy individuals who are at risk of poor outcome. For example, if someone has had severe viral infection and you're choosing how to treat them with any sort of drug, if you know that person's viral infection, a chronic history of viral infection or liver disease, has given them very short telomeres, you wouldn't want to choose a therapy that's going to demand high proliferation, is going to be toxic to the normal cells. Whereas if your patient, in fact, has a very large buffer, a large reservoir of telomere length, then you can do a very harsh treatment that will ablate even some of the normal cells, and the remaining normal cells will be able to repopulate. Another purpose would be to determine individuals who are in need of early intervention lifestyle modifications. We all have to choose how much care to take with our health as we get older. We realize that a bit more. In avoiding things like oxidative damage, in avoiding inflammation. And I think your choice of lifestyle can be influenced by how much specific factors are really going to compromise your tissue renewal. Again, if you were genetically blessed with huge telomere length, perhaps you don't have to worry about that long distance running. But if not, maybe you should think about changing your exercise regimen. So another purpose that I really think hasn't yet been executed, but should be on the plate tomorrow, is to select donors and populations of cells that are most suitable. So early in life, we lose an enormous quantity of telomere length as our immune system establishes itself. And we lose less as adults. But when a bone marrow transplant is done into hypocellular marrow, those transplanted cells, they have to divide like you were young again. They have a huge task of repopulation. And in fact, that huge task is it has been demonstrated, tax on telomere length. So if we tested donors and particular cell populations before transplant, we might have a much better success rate in long-term repopulation if we use cells with longer telomeres. And I really hope that that is considered seriously. So one last slide. You're convinced You're going to look in the future for new developments in telomere testing, what to consider. I've told you we're getting there. There's much left to do. There will be great improvements in the next few years. But one point I need to make is that this test, when you do it, needs to provide not just an average telomere length of a whole cell population. It must be smarter than that. You have to be aware, unlike for a genotype test, that different cells in the population will have different telomere lengths. There's a difference between a telomere test and a gene test. And of course, you want it to be accurate and usable. So I will thank you and again encourage you to come to the exhibition hall and ask me any additional questions, to think about monitoring telomere lengths at a telomere by telomere resolution. And as a scientific, sorry, we'll go back. Nope, we'll go forward. Up. Yeah, they put it here. Okay. I'm a consultant for one of these telomere length companies, Life Length. They have a booth here. But in addition, I think that where the telomere tests are now is just very beginning stages, and we need feedback from all of you on how to develop these tests in the future. So thank you for your attention, and I'll stand for the next speaker.